Assamualaikum and greetings to my examiner, Dr. Norshida, and my supervisor, Dr. Zarizal. I am Siti Nur Balqis Binti Mohd from ISMPKH Semester 6. My title for progress presentation is Prevalence, Antimicrobial Susceptibility Patterns, and Pantone-Valentine Leukolysin of Staphylococcus aureus isolated from nasal swab of goats and sheep in ladang pasir akar. These are the content that I'll be presenting today. For the introduction, in short, it's about the serious problem that S. aureus can brought into our population and the dangerous mechanism of antibiotic resistance as well as the negative effect of PVL genes. 4 problem statements. First, S. aureus has been main clinical livestock pathogen worldwide. Second, the presence of antimicrobial resistance of S. aureus has reduced the effect of antimicrobial agents. The main significance of studies are to determine the pattern of antimicrobial susceptibility and the presence of PVL genes in goats and sheep in ladang pasir akar. Research objectives in these studies are first to determine the prevalence of Staphylococcus aureus from nasal swab of goats and sheep. Second, to classify the pattern of antimicrobial susceptibility for each isolated of S. aureus towards various antimicrobial agents. Third, to investigate the availability and prevalence of Pantone-Valentine Leukolysin genes from the isolated S. aureus. For materials and method, it has been approved by UFRAT under 04/009. For methodology, first, nasal swab sample of goats and sheep was collected and brought into microbiology lab for bacteria isolation using bread pakar agar (BPA). The suspected colony was continued for phenotypic identification using selective media, which are PBA, and the formation of lecithinase. For the genotypic identification using PCR, which it was targeting the NUC gene and PVL gene. The confirmed S. aureus species that we have identified was carried on with antibiotic susceptibility testing using this diffusion method. Lastly, the data will be conducted using descriptive analysis, which is in the form of percentage to show that the prevalence of S. aureus. For result and discussion, we have found that 50.6% has confirmed positive for S. aureus isolated from nasal swab sample, which total overall 137 samples were positive SA out of 271 samples, while the remaining are negative results. For phenotypic identification, we have identified the colonies are confirmed SA by observing clear zone around the black colonies when the swab samples were stripped on bread pakar agar. Yellow colonies were observed when the sample stripped on nutrient agar. This bar chart shows the percentage of antibiotic susceptibility by S. aureus. The purple color represents percentage of sample were susceptible to the drugs. Blue color indicate that the sample resistance, and the orange color is the percentage of intermediate. 45.5% of the sample were resistant toward cephosity. This figure is the result obtained from gel electrophoresis using PCR method. It has interpreted that all the samples were negative in consisting PVL gene. What I can conclude is that the prevalence of S. aureus is high in small ruminant nasal swab samples compared to previous studies. Also, high prevalence of S. aureus isolated in LPA were resistant toward cephositin. And lastly, there were no isolated were detected in PVL genes, which shows that the ruminants are free from skin infection. Finally, for my path forwards, these are the several things that still in progress for my current research studies, which are data analysis and writing, thesis writing that need to be submitted on the 9th June, and the poster presentation. These are the references that I use in this presentation. That's all from me. Thank you for listening.